이 논문의 핵심을 살펴보겠습니다.
Welcome, everyone. Let's just jump right into it, shall we? Today, we're diving deep into the core of some truly fascinating research. We're going to unpack the intricate defense network of our cells, essentially looking at this explainer as a masterclass on how our biological power plants protect themselves from a total meltdown.
So here's the million-dollar question. 미토콘드리아가 고장나면 세포는 어떻게 생존할까요? How do cells survive when mitochondria break down? It's a massive biological puzzle. I mean, imagine a giant, high-output power plant that suddenly faces a catastrophic internal failure. If they don't shut things down just right, you get an absolute, uncontrollable meltdown. Well, our cells face that exact same danger every single day. So how does this microscopic power plant prevent a complete disaster when things go wrong? Let's figure it out.
Section 1. The integrated mitochondrial network. This is where we see a monumental shift, taking years of isolated research on CHCHD2, CHCHD10, and OMA1, and combining it into one master blueprint. Now, this completely flips our old assumptions right on their head. The old view? Well, we used to think the emergency enzyme OMA1 was pretty simple. We assumed it just woke up on its own whenever the membrane voltage dropped. But the new view is way more interesting. It reveals this hidden, highly organized hierarchy. OMA1 isn't actually the top boss calling the shots. Instead, the CHCHD2 and CHCHD10 complex acts as the ultimate upstream boss. They're basically the master safety inspectors, actively holding OMA1 back and suppressing it.
Moving on to Section 2, the peaceful, normal state. Let's picture a healthy, well-oiled cellular machine completely at peace.
Think about the normal day-to-day operations at our biological power plant. You've got CHCD2 and CHCD10 bound tightly together, working as this super efficient team. And what are they doing? They're actively holding down the cellular emergency switch, which is OMA1. So as long as this dynamic duo is in charge and functioning properly, that emergency shutdown switch is locked safely in the off position. And because that switch is securely off, our normal state checklist looks great. First, OPA1 maintains those stable internal folds, or cristae. Second, the DELE1 protein, it remains perfectly intact and uncut. And third, out in the wider cell, protein synthesis, the manufacturing line, is humming along normally. Everything is stable, structurally sound, and energy production is just running smoothly. Section 3, stress and the awakening of OMA1. Because, peace never lasts forever and our power grid is about to drop. The catalyst for this crisis is a chemical called CCCP. In the lab, researchers need a reliable way to simulate a power plant failure, right? So CCCP is the go-to stressor used in these studies. What it does is force the electrical voltage of the mitochondrial membrane to just completely collapse. It induces what we call depolarization. So when that voltage drops due to stress, crisis hits. The CHCHD2 and CHCHD10 guards, they literally lose their grip. Their protective complex fails. And the very second they let go, the suppression is lifted, and OMA1, our emergency switch, finally awakens. The biological alarm bells are officially ringing. Which brings us to Section 4, mitochondrial emergency restructuring. Let's unpack the immediate emergency protocols OMA1 initiates now that it's awake. OMA1 triggers multiple pathways, basically all at once. Pathway 1 is a physical structural shutdown. Activated OMA1 acts like a pair of biological scissors, cleaving a structural protein called long OPA1 into a shorter form. This intentional cutting actually causes the cristae, those internal folds we talked about earlier, to fragment and break apart. It's wild. The cell is literally dismantling its own factory floor just to contain the damage and prevent a larger catastrophe. And while the floor is being dismantled, Pathway 2 sends out a massive distress signal. Step 1, OMA1 cuts the DELE1 protein, and this shortened piece actually physically moves out of the mitochondria. Step 2, it activates an enzyme called HRI. Step 3, that triggers EIF2-alpha. And finally, Step 4, it activates a major transcription factor called ATF4. Boom. The alarm signal has successfully reached the cell's main headquarters. So what exactly does ATF4 do with this blaring alarm? It drastically decreases cellular translation, effectively ordering the cell to stop making new proteins. Now, you might be wondering, why on earth would a cell stop making proteins during an emergency? Well, think about it. If your factory is broken and literally on fire, sending in brand new raw materials is only going to make the disaster worse. It halts production, so the cell isn't overwhelmed by building materials it has absolutely no energy to process.
Section 5. The ultimate protective shield for survival. Let's tie all these discoveries together and look at the real ultimate purpose here.
Because here is a massive recent plot twist from a newly published paper. The missing link is a protein called DNA JC15. Activated OMA1 doesn't just dismantle the inside and halt the factory outside, it also cleaves DNA JC15 to literally lock the front doors of the mitochondria. It restricts new proteins from even being imported in. It is the final puzzle piece. Complete lockdown. Absolutely no shipments are making it past that door.
If we look at this comparison, the stark contrast is what defines survival. Under normal conditions, you got stable cristae, inactive OMA1, a normal protein synthesis. But throw in that CCP stress, and the whole system flips right into survival mode. Cristae fragment, OMA1 goes highly active, and protein synthesis tanks. It is a perfectly, beautifully synchronized response.
And here is the absolute key takeaway I want you to remember. This elaborate shutdown, it isn't just random cellular damage. No way. It's a highly sophisticated survival strategy. By doing all of this, the cell achieves 3 massive goals. It reduces precious ATP energy consumption, it drops the levels of harmful reactive oxygen species, or ROS, and it maintains proteostasis to stop toxic proteins from building up. This exact protective shield is what helps prevent terrible neurodegenerative diseases like Parkinson's and ALS.
Which leaves us with one final, pretty provocative thought. Is cellular aging really just unavoidable wear and tear? Or could unlocking this CHCHD2/10 and OMA1 master switch actually be the key to rewriting the rules of survival entirely? The microscopic intelligence inside ourselves is literally breathtaking, and this unified model proves we are just scratching the surface of understanding its true power. Thanks so much for joining me for this explainer, and definitely keep exploring.